Okay, so this is the answer booklet for the Triple Biology Higher Tier 2019 paper. The image shows human chromosomes. State the number of pairs of chromosomes present in a single body cell. So you got 1, 2, 3, 4, 5, blah, 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 all the way up to 22. And then we have the two sex chromosomes, one for a male and one for a female. So we have 23 pairs in total. State which sex chromosome are present for a man, and it's XY. And then for a female, it is XX. Complete the Punnett square below to show the sex chromosomes in the gametes of a male and a female parent in their possible offspring. So the male XY, female XX, make sure they are the correct way around or you won't get the marks. And then 50% a girl, 50% a boy. Height in humans is controlled by many genes as well as by environmental factors. Females usually reach their full height by 18 and males by 25. Students investigated the heights of male and female airline cabin crew. All cabin crew must be between 1.58 and 1.9 metres in height and between 18 and 45 years of age. The students said that they expected the males to be taller than the females. They collected their data by selecting 7 males and 7 females at random and asking them to state their heights. They gave their heights in feet and inches, and the students converted their data into metres. The results expressed in 2 decimal places are shown in the table. The next question asks you to calculate the height of the females, so you add them all up and divide by the 7, which gives me an answer of 1.76 for that question. State the hypothesis of the students. It was that they said that the males are taller than the females. State whether the results of the investigation support their hypothesis. And it shows, though, that the females' average height is greater. The females is 1.76 and the males are 1.74. So it doesn't support their hypothesis. Give one way in which the strength of the evidence could be improved. They could increase their sample size. State one source of inaccuracy. They requested the heights, they didn't actually measure the height, so they could be lying or got it wrong. Cystic fibrosis is a serious medical condition which affects the lungs. It results from a mutation in the DNA of a single gene, which gives rise to a recessive allele. From that then, explain the meaning of the term recessive allele. It's a form of a gene which only shows when 2 recessive alleles are present in an individual. The diagram below shows parts of the family tree of a family which has one member whom has cystic fibrosis. State the effect of the mutation in DNA when it's a change in the DNA structure. From the family tree, state the number of 2 children who are known to be heterozygous. So heterozygous, when you are looking, it's when the alleles are different. When the alleles are different, then we've got to work it out. So if you've got a child who is recessive, they're going to have 2 lower cases, such as little n, which they inherit half from mum, so 1, and then the other one from dad, etc. Okay, so both parents we know need to have a little n each because they've given it to their child. Then we know that they haven't got cystic fibrosis, so they can't have 2 lowercase n's. If they did, they would have it. So they have to have a big N as well. So parents 1 and 2 are heterozygous. Suggest what advice the genetic counsellor would give individuals 5 and 6 about the chance of them having a child with cystic fibrosis. So a family with DNA testing shows they haven't got cystic fibrosis. So we know that that male is going to be big N, homozygous dominant. They haven't got it in their family tree. What they will have then, if they have kids, no child's going to end up with cystic fibrosis because the mum is always going to pass on a dominant allele, never a recessive one. So no children are going to have cystic fibrosis because the female hasn't got any recessive alleles to pass on. Cystic fibrosis is treated by intensive physiotherapy, which can be very stressful. Medical scientists are now working to develop an alternative treatment called gene therapy. This process is summarised in the flow chart below. Lipid capsules are introduced into the lungs, state how this would be done, and it is done using an inhaler. In order for this therapy to be successful, 2 further difficulties must be overcome. Describe these 2 difficulties and suggest why this therapy is not permanent. And it's because the genes need to enter the lungs. They have to enter the DNA and function, but it's not a cure because the lung cells eventually die and constantly reproduce by mitosis. What is meant by a sense organ is an organ which responds to a stimuli like light, sound, heat and pressure. For example, name the 2 components of the central nervous system is the brain and spinal cord. So 3 properties of a reflex action: they're fast, automatic and protective. And now we have to label a receptor and a motor neuron. So our receptors are on the end, okay, they detect the stimulus, then our motor neuron takes it back to an effector. Motor neuron disease is a rare condition that's caused by the motor neurons not functioning correctly. Suggests the effect it's going to have on the reflex action. It's going to slow it down. It's not going to get the effector to withdraw their hand or to have the response as quickly as we would want. Chlamydia is the most common sexually transmitted disease diagnosed in Wales. In 2013, there were 7,076 reported cases of chlamydia, and by 2014 the number had risen to 5,452, of which 71% were 16 to 24-year-olds. Give the scientific name of the pathogen that causes the disease, so it's Chlamydia trachomatis. State the name of the group of microorganisms to which the pathogen belongs. It is a bacteria. Describe how the spread of chlamydia can be prevented using condoms during sexual intercourse. Calculate the number of 60 to 28-year-olds diagnosed with chlamydia in 2014. So 2014, we had 5,452 and it was 71%, so times 0.711, and the number of young people were 3,876. Calculate the percentage increase in the new chlamydia diagnosis between 2013 and 2014. So the difference was 376 divided by the original in 2013, which is 5,076, times by 100. So the percentage increase is 7.41%. So just like public health awareness campaigns targeting the spread of chlamydia are important, because there's an increase in the number of young people getting chlamydia, which can lead to infertility in babies. There are many different species of snails. Students were asked to estimate the population density of the garden snail in a local park using the following method. So, use a tape measure to mark out an area, search for the snails in the marked area and pick them off. Use an identification key to identify the garden snails. Count the number of garden snails caught and mark their shells. Allow the nail varnish to dry. Search the marked area again after a week. Count the total number of garden snails collected. Release the snails and estimate the population size using the equation. So the results showed that the number of garden snails in the first sample was 198, in the second sample 152, and the ones that are marked were 8. Garden snails are invertebrates. State what is meant by the term invertebrate. So it's animals without a backbone. Make sure you put backbone and not spine. You won't get the mark for spine. Explain why it's important to use a scientific name to identify and classify organisms. It's because it's universal and avoids confusion. You only need to put that. Do not write anything else. Use the formula given opposite to estimate the population of garden snails in the area sampled. So we have to do 198 times 152, which gives me 30,096, and divide it by the number marked using this equation over here, which gives me an answer of an estimate of 3,762 snails in the field. Use your answer from part B to calculate the population density per metre squared of the garden snails. So the area was 30 by 30, so times that to get 900 metres squared. And then the population 3,762 divided by 900, and then per metre squared we would have 4 snails per metre. So 2 assumptions made when using the method given on page 12 to estimate. So we assume there's been no deaths or births, and there's no migration such as emigration or immigration into the field. Suggest how students could improve the strength of their survey. They could increase the sample area. Suggest 2 precautions the students should take in using the survey to reduce the risk of harm. So to not litter and to avoid trampling on the snails. The diagram below shows a section through the human brain and also the excretory system. On the diagram of the label, the aorta, I've missed, and the medulla of the kidney. So the aorta is going towards the kidney because it's going away from the heart, and then a medulla is there in the kidney. Okay. Next question. Explain the link between the quantity of antidiuretic hormone produced by the brain and the volume or concentration of urine produced in a person who is dehydrated. So first thing, the brain is going to monitor your water levels. If the water levels in the blood are low, the blood is going to be concentrated. With that then, more ADH is going to be released into the blood. As we've got an increase in ADH, we're going to have more reabsorption by the kidney back into the blood. What's going to happen then, the volume of urine will decrease because we're going to absorb more water back, so the urine becomes more concentrated. The article below was taken from a newspaper in early 2017. DNA profiling snares Welsh sheep rustler after investigation. On January 15, a number of pregnant sheep were stolen from a farm. Inquiries within the local farming community revealed a pregnant sheep which was suspected of having been stolen and been sold at a livestock market. The Animal Plant Health Agency wanted to carry out DNA analysis on the sheep and its lamb. They had to wait 2 months for the sheep to give birth before taking a sample of blood from the lamb. The diagram shows the genetic profile of the ram, the suspected stolen sheep and the newborn lamb. Describe the process of genetic profiling. So it's cutting DNA which are separated into bands for true marks. Explain how the genetic profile provides evidence that the sheep had been stolen. And if we look, it's because half of the DNA comes from the ram. So if you look at the newborn sheep, that matches with that one. And then we've got that one matches with the top one, not both of them. And then the stolen sheep, then if I do it in a different colour, has the other half that match the bottom half. Okay, so half the lamb's DNA comes from the ram, the other half comes from the sheep, so showing the lamb is related to both of them. Remember, we get half our DNA from mum and half from dad. So one possible health benefit and suggest one ethical concerns linked to DNA profiling. So the benefit, we can identify any diseases, and ethical concern, we have privacy concerns that is on file forever. Staphylococcus aureus, it's a bacterium which is a common cause in infection in human wounds. It's become resistant to antibiotics. Honey has been used as a wound dressing for thousands of years. Scientists carried out a laboratory investigation to study the effect of honey on the growth of this bacteria. A series of agar plates containing different honey concentrations were prepared. A culture containing the bacteria was transferred to each of the agar plates. And then we have our results below. The plates were incubated for 24 hours and then inspected for the presence of bacterial colonies. The results are shown below. So the concentration of honey, when we get to 3%, we have bacterial colonies. After 3%, we have no bacterial colonies. State the link between the number of bacterial colonies present on the agar in diagram 3 at 24 hours and the number of bacteria in the original sample. We assume that one bacteria forms one colony. So if we have 10 colonies, we assume that 10 bacteria started. Suggest the temperature at which the agar plates would be incubated, and it would be between 36.5 and 37.5, and it's because they grow best at body temperature. Take 2 basic aseptic techniques that should be followed. So sterilise the agar plate and seal the lids with tape so that it avoids cross-contamination. Using the diagram to calculate the number of bacteria present at 1 centimetre cubed of the original sample. Okay, so we do 22 divided by 0.0003, which gives an answer of 7.3 times 10 to the power of 4. Using the table on page 20, state the minimum number concentration of honey required to stop the growth, which would be 3.5, because after 3 it decreased. So there was none there. Suggest how scientists could develop their investigation to get more accurate value for the minimum concentration of honey required to stop the growth of bacteria. They would use a range of concentrations between 3 and 4, so like 3.2, 3.4, 3.6, 3.84, for example, to work out exactly when no bacteria grew. State the 3 preclinical stages that would be carried out in order to develop honey as the new medicine. So they would test on cells in labs, then they would test on animals, and then they would test on healthy volunteers. The eye is a complex organ made up of many tissues. Diseases of the eye occur when one or more of these tissues are damaged or stop functioning. Stem cell treatments are now being developed and used to treat many eye diseases. So, example, when repairing of the cornea, this treatment is now licensed for use. Stem cells at the edge of the cornea replace cells that are consistently being worn away. These stem cells may also be damaged by burns, radiation, genetic diseases, infection and drug use. The diagram below shows stem cell treatment to repair the cornea. This treatment uses stem cells from a healthy part of the cornea of an affected individual. Then we have repairing the retina, and then age-related macular degeneration in the retina stop functioning. The diagram below shows stem cell treatment that uses the embryonic stem cells. So State the function of the cornea. The cornea allows light to enter into the eye. Explain the meaning of the term stem cell. So it's an undifferentiated or non-specialised cell, but it can develop into any type of cell that we need it to. State the name of the type of cell division that occurs in the petri dishes in diagram 1 and 2 and explain the significance. And it is mitosis, and it's going to produce new cells, which you want new stem cells constantly regenerating, that are genetically identical to the cells that we need. Suggest why example 1 does not work if both eyes are badly damaged, because there's going to be no stem cells available to take from the person. Say 2 advantages of using stem cells as a treatment in example 1 compared to using sample 2. Well, it's coming from the same person, so we're going to have no donor required. We're going to find someone. With that then, because it's cells from the person, there can be no rejection because there's not be any foreign antigens, and there's no ethical issues with destroying embryos after getting the stem cells from them.